What I hope to do today is to tell you about one of the things going on in my group where we've been seeking to use the capabilities of smartphones in biosensing. And so this project got its start partly because I'm a gadget enthusiast, and so today I'm going to do a little experiment because I'm an experimentalist. I'm giving my talk on my smartphone, and I'm going to try to advance the slides with my Apple Watch. So we'll see how well it works. But really, when the iPhone first came out, I was one of the first ones to sign up. I have to admit that I was a fan. And not long after that, I had met with an alumni from Illinois that came back here to share his experiences making a fortune, being one of the first people to sell smartphone apps that were games. And so he was showing me this, like video game that he made where you could hold up the phone and the video camera would take a picture of the outside world, and you could pretend by pressing buttons on the phone screen that you were shooting missiles, at the person across the table from you, or at the thing across the street or whatever. And so, at $1 per app, he sold like millions of copies of the app. And I remember thinking, that's really great, but certainly there must be something more interesting or more useful you can do with a smartphone than fire virtual missiles at your friends. So since I work on biosensing, I thought, well, why not use the internal sensing capabilities of a phone to do biosensing measurements? And it kind of makes sense if you think about it, because phones have cameras in them. The cameras turn out to be getting better and better every year. So like a 4-megapixel camera was it was pretty good, like in 2007. But now we would throw that phone away, and now they have like 10 megapixels, 12 megapixel cameras. The other thing is that the phone's cameras have gotten pretty good in their ability to detect low light. So people like to be able to take pictures indoors of their friends, selfies, and all that stuff. But since the cameras sensing pixels have gotten pretty sensitive, we found that those are pretty good for detecting biological things as well. So what I'll tell you about today are some projects we've been working on in my group the last couple of years. This all started with a few undergraduate students for like a senior design lab. I went to the class and said, does anyone want to work on a smartphone biosensor? And 5 kids raised their hand and said, yeah, let's try it out. The first semester we tried to build something. Didn't work at all. It was a total disaster. It looked like a lump of coal. We ended up, the next semester there was a student who was really great. There's an undergrad student, ECE student, who really stuck with it and helped us design something in a more methodical way. Machined it out of aluminum, and it worked really well. They got our first results. And then we've gotten several NSF grants based on this, to expand it. And I'll tell you about some of our plans going forward. So let's. Oh, I got to do it this way. Here we go. So, what I hope to do is tell you why you want to use mobile sensors for biosensing, and I'll share with you 3 different technologies, actually 4 different technologies that we've developed that use the internal sensing components of phones. All right, so a lot of the what I call the grand challenges in food and pharmaceutical safety involve sensing things. And so if we think about detecting pathogens in food that we're about to eat, detecting pathogens in animals, such as the horses, cows, pigs, chickens that can spread diseases amongst themselves. Pathogen sensing in people, also very important for trying to contain outbreaks of things as routine as the flu or as serious as Ebola. Detecting bacteria in various places, very important. Detecting allergens in food is another area where people are interested in being able to detect things in their food they're about to eat. Or for companies that are manufacturing food, detecting contaminants in the ingredients that they're about to incorporate by the ton into cereals, baby formula, things like that end up getting shipped all across the world. People are interested in detecting things in the context of agriculture. For example, detecting fungal toxins in grain that's stored in silos. But if fungus starts to grow, the fungus produces toxins that renders that grain inedible, even by animals. The only thing they can do with that grain, if it has a toxin in it, is to make it into ethanol. And so, this is very important in the farming community. And then, of course, detecting contaminants in food and pharmaceutical products, whatever they may be, in terms of food or drugs. I'll show you some applications detecting people doing unethical things to drugs that people will take, trying to save money, make a little more profit, unscrupulous people that have to be countered with biosensing. So in all these applications, you can do all these measurements by taking a sample, sending it off to a lab, having a technician in the basement of the hospital or in some testing lab in some part of the country do a test and send you the results back, where you might get them a day or 3 days or a week later. People are interested in getting a measurement like right here, right now. To measure the food that's right in front of them, to measure the animal that's right in front of them, to measure the silo of grain right there, to measure the stuff on the shipping dock of the company, so that they can move forward with their eating, diagnosing an animal, manufacturing things. I keep pressing the wrong button. I have to get used to this. In addition to detecting things like that, we have detecting things in the environment. So I'll just kind of mention these a little more quickly. For example, detecting things in soil and farmland, pesticides, insecticides that might be on our food. Also, things like rootworms or fungus, also important to do outside. Detecting the contaminants in water resources, such as rivers, groundwater, the beach, where beach has to be tested periodically for bacterial contamination like E. coli. And then, of course, there's applications that the military is concerned with, such as attacks by the bad guys with chemical or biological weapons. And so the troops may need to be very concerned that they'll be exposed to something. They have to gown up or move and be able to take measurements right where they are, not send a sample off to the lab and wait, everyone will be dead by the time they get the results back. And so what we're seeking to do in our work is to take several different sensing technologies that are used in laboratories. And I show them here. The first one is label-free optical biosensing technology called a photonic crystal sensor. Second one is a enzyme-linked immunosorbent assay reader. Third one is a fluorimeter. These are all things that work just fine. People have them in diagnostic laboratories, but each one of them on its own weighs like tens of pounds. So we're kind of talking about a total 150 pounds of laboratory instruments here, and it can cost tens of thousands or thousands of dollars. And so what we've been doing is taking all three of those and putting them in one multi-mode sensing instrument that uses the internal sensing capabilities of, in this case, our first one was an iPhone 4 that has less than $1,000 of components in it and weighs less than 2 pounds. And so this is what we were seeking to do. So we know how great smartphones are. I don't even have to tell you that anymore. And that not only have their computing and imaging and sensing capabilities gotten better and better, they keep coming down in price and are used by all people. I can't even remember how many billion smartphones have been sold now. There's still probably a few people left that don't have them if they're under 3 years old or something like that. So what we're seeking to do is to use the internal capabilities in a phone to do all sorts of work. And so the first example I'll show you is what I call a label-free optical biosensor. And this is using a sensing structure developed in my group and then commercialized by a company that I founded using a structure called a photonic crystal. I won't say too much about photonic crystal except to say that it's a nanostructured surface that we can make out of plastic and apply to like a glass surface, like a microscope slide, and cover this whole surface with the photonic crystal. The basic property that the photonic crystal has is that if you shine all wavelengths through it, only one wavelength will be reflected. And since that wavelength gets reflected, you'll see a sharp dip in the transmission spectrum of light that comes through the photonic crystal. So if we shine white light at the photonic crystal, one narrow band of wavelengths gets reflected back at you, resulting in a dip in the transmission, and you can measure that with a spectrometer. So our first thought was, how could we use a smartphone to act like a spectrometer and to measure optical devices like a photonic crystal. And so this is a thing I told you about where the intrepid student, his name was Dustin. I tried so hard to get him to come to grad school, but he wanted to go make money in the real world, so he took a job at IBM instead. I still am mad about that. But anyway, Dustin was responsible for designing and getting this device made out of machined aluminum at our machine shop in the ECE department. What it does is take light from a broadband source, such as a light bulb, or you could hold it up to the sun. But the broadband light enters a pinhole, so just a small hole. The light that comes through the pinhole is mostly collimated, but we put a collimating lens in the path to collimate it even more. We pass that light through the photonic crystal. There's a little slot in the back of this cradle that the photonic crystal slides in. And then the light that comes through the photonic crystal is focused along one axis onto a diffraction grating. And the key thing is this diffraction grating that's just sitting right on the back of the phone's back-facing camera. And so the thing that the diffraction grating does is to take the components of the light and to spread them out along the pixels of the camera. So instead of getting a picture, you get a spectrum of whatever light is entering this diffraction grating. So that's it. It's not super complicated, but this whole thing has about $200 worth of components in it. If we don't cost the aluminum, which probably costs like $1,000 to get the machine shop to make it. Okay, so this basically is made to do the same job as this 60-pound photonic crystal detection instrument. This instrument, it's not totally fair. This thing is meant for doing high throughput measurements and measuring photonic crystal biosensors where they're in these microplate formats that can do like 384 or 1536 measurements at once. But in our case, we were thinking of mobile diagnostics measurements at once. So we've incorporated our biosensor into a little plastic cartridge, about the same size as a microscope slide. Okay, and so what do you get if you use your smartphone to measure a spectrum like this? And so here's what the spectrum looks like, spread out across the pixels of the iPhone 4's camera, going from blue to red. If we take our photonic crystal and put it in the optical path, remember some of the wavelengths get reflected back, so they don't make it through, and so we see this dark band in the spectrum because those wavelengths were subtracted away by the photonic crystal. And so if we measure the transmission spectrum, this is someone else's phone, not mine. You know who this is? It's a cool song. Someone left their phone up there. It's not mine. Okay, so if we have the photonic crystal in the optical path, we get this dark band. These are the wavelengths that are subtracted out. And so if we measure them with a spectrometer, these wavelengths are in the visible part of the spectrum. You can kind of see they're in the green band of the spectrum. If we just use the pixels of the phone and look at this dip in the spectrum, we see exactly the same thing. And so all we need to do is to turn this into a spectrometer, is to somehow let the phone know which pixels in the image correspond to what wavelength. And so we did this by taking a green laser pointer and a red laser pointer and shining them into the pinhole. And we see high intensity peak for those laser wavelengths. We know exactly what those wavelengths are. And so now we can calibrate our phone spectrometer to any wavelength in between. So what this shows is the light output from a tungsten lamp, so an incandescent lamp, measured with a actual spectrometer. Here it is measured with the iPhone. And so you notice there's this little shark fin characteristic. Doesn't follow the actual spectrum from the light bulb. This is because the pixels inside the camera and the software that processes the information puts some bias on some wavelengths more than others. And so this turns up in our color photos and things. Maybe it makes our skin tones look better, or our clothes look more brilliant or something. I don't know. But these characteristics are in the spectral data. And so once we know that, though, we can calibrate that out and then get a more accurate spectrum for anything else. And so one of the students was a software engineer, took a class on how to make an app. His first app was an app for measuring smartphone biosensor spectra. So you could enter in some information and measure these transmission dips and measure the wavelengths that the dip occurs. So how do we use this for biosensing? So we did this like several different applications, but let me show you one that's an important one. And so we were interested in developing a test for HIV viral load monitoring. And so we have people that have an AIDS virus that are taking antiretroviral therapy have to measure their viral load frequently so that they can make adjustments in the drug cocktail that they're taking to manage the virus. And so since this is a test that has to be done frequently, it would be best actually if the patient didn't have to go to the hospital every time, draw blood and do a test performed by polymerase chain reaction, kind of an expensive test. What we're seeking to develop is a mobile test, a rapid test, something that could be done inexpensively and accurately. So what we did was to prepare our photonic crystal with antibodies that recognize the proteins that are displayed on the outside surface of the HIV virus. And so we can prepare our sensor with these antibodies. They're attached there permanently with chemical bonds. And so the way our photonic crystal works is that we can measure I guess here I showed the reflected peak, or we can measure the transmission dip, but here I showed peaks. We can measure shifts in the reflected wavelength of the photonic crystal when biological stuff attaches to the surface. And so when virus particles get attached here, we would measure a change in this wavelength, and then we would seek to measure that change in wavelength using our phone spectrometer. And so this just shows the changes in wavelength. We call it the peak wavelength value, or the PWV. We can measure the change in the peak wavelength value as we attach the antibody, as we do a blocking step with bovine serum albumin, and as we attach the HIV virus. On the actual test, actually, the sensor will be prepared with all these steps up to the BSA. Then to perform the test, a droplet of serum goes on the sensor. It incubates there for 30 minutes. And then we measure the wavelength change from before the HIV was added to after. And so we did these tests on serum. Actually, we added the HIV. We had serum not from HIV patients, but we added the HIV at a range of concentrations going from 10 to the 5th per ml to 10 to the 8th per ml. And here just showing the change in wavelength that occurs. Just to give you an idea, here's the photonic crystal biosensor structure, and there's one HIV virus particle. So they're small. We're only capturing a few virus particles per square millimeter on these. I guess about 100 per square millimeter, if I recall correctly. And so yet we're still able to measure wavelength shifts on the order of like fractions of nanometers using a smartphone instrument like this. So I wanted to show you another application. This is for something called enzyme-linked immunosorbent assay, or called ELISA for short. The ELISA assay is a really important one for diagnostics because it's like the most commonly used diagnostic test for any kind of disease. There are ELISA kits for every cancer biomarker, every cardiac biomarker. There are literally thousands of ELISA tests. But their methodology is all the same, where you prepare a surface, such as in a microplate, with a capturing antibody. The antibody gathers the antigen, the thing that's detected, out of a test sample. And then there's a second antibody called, amazingly, the secondary antibody that binds also with that detected analyte. And so it makes a sandwich. So we kind of have like bread, and like turkey. I guess I'm getting hungry in the middle here. And so this is called a sandwich assay. The top part of the sandwich, the secondary antibody, has an enzyme on it that reacts with a substrate to generate a colored product. So to read out the result of an ELISA assay, what you do is you wait for the colored product to develop. It changes the liquid on top of the assay from colorless to purple, and the amount of purple tells you quantitatively how much analyte was present in the sample. And so this is a process that has a few steps in it, but is performed all over the world for doing diagnostics. And the basic readout is measuring change in color absorption. So this is very similar, actually, to our photonic crystal situation, where our photonic crystal was subtracting out a very specific band of wavelengths. But if we measure like the absorbance of purple-colored liquid, we could look at one wavelength and see how much darkness develops, and measure the absorption as a function of concentration. So we could do a test like this the following way. Using that exact same hardware, we could take a broadband light source, pass it through our test sample, the thing that's turning purple. So it goes through our pinhole, goes through our collimating lens, goes through our diffraction grating, and then now the camera can measure the absorption spectrum of that test sample. And so this is what it looks like. And so here we were doing a biomarker test for an ELISA test for a biomarker called interleukin-6, called IL6. This is a common biomarker used for cancer diagnostics. So what this shows is the absorption spectrum measured with the phone as a function of IL6 concentration going from low concentration to high concentration. So this is really, what it really means is that the solution is getting more and more bluish purple with the higher concentrations of IL6. So a typical ELISA reader will actually only be capable of looking at one wavelength, but since we have the whole spectrum, we can actually look at all the wavelengths. And what we do is to integrate the area under this curve to give us a final absorption number. And so this shows the transmission efficiency or absorption. I'm sorry that the scales are kind of flipped, as a function of concentration of interleukin-6. This is where we're adding interleukin-6 intentionally at known concentrations, but it's just showing that we can get a nice dose-response curve, and that our limits of detection were around 2 picograms per ml. Measuring the same exact liquid in a official laboratory ELISA reader, our limits of detection were 1 picogram per ml. And so basically we could get the same detection sensitivity for performing an assay, even when we use the phone for doing the readout. I wanted to show you another example just because it's fun and it's before lunch, and so but involves, but also important to people with allergies, is measuring peanut allergen. And so first let me ask if anyone here has like severe peanut allergy. Because there's always at least like 1 or 2 people, and it's becoming more and more common, not just for peanuts, but also for like shellfish. Even I've met people with severe allergies to soybean products. But what these things have in common is that there's some protein, actually there's a set of proteins that people develop allergic reaction to. And then these allergens can be detected with an ELISA assay. And so what we've done here is just to perform a ELISA assay for peanut allergen in various types of cookies. And so we took cookies, we bought them from several sources. We take a sample of cookie, mix it with hot water for a few minutes. We centrifuge the stuff so the cookie particles go to the bottom, and we do our ELISA test on the supernatant liquid. But then here's some of the results. And so we performed testing on cookies with peanut allergen at various concentrations using the smartphone and using the ELISA reader. These are just 4 concentrations, but again, we measure a nice dose response curve. And then we followed this up by measuring like the unknown peanut allergen concentrations in cookies we got from various places. And so, for example, if you take peanut butter cookies, they've got tons of peanut allergen in them. They're off the chart. Actually, they went over the maximum level. But if you take Oreo cookies, for example, no peanut allergen in them. The grocery store in town, Schnucks, they make sugar cookies in a separate peanut-free facility. So we wanted to test them out. So they were below the detection limit, or our negative control actually for peanut allergen. But there's a bakery in town called Insomnia Bakery that makes the most delicious cookies. If you want to find some great cookies this weekend, definitely go there. They're open all night, which is one of their benefits. But if you have peanut allergy, don't get the sugar cookies because they make them in the same bakery that makes the peanut butter cookies, and so you can find plenty of peanut allergen in their sugar cookies. So this is the kind of test that you're granted. If you had to wait 20 minutes to perform this test before you could eat your cookie, I couldn't do it. But we're developing methods to make these kinds of tests more easily performed by a consumer and more rapid. Another application I wanted to show you was how to use this thing as a fluorimeter, in other words, detecting fluorescence. And so here's the competition, the Agilent laboratory fluorimeter. Here, all we do is take a green laser pointer, so a $5 laser pointer, focus the light into the center of a liquid test sample. The fluorescence emission comes out everywhere, but we put a lens on one side and then collect some of the light into an optical fiber. So then we take the output of our optical fiber and put that into our little pinhole that interfaces with the camera. And so now, instead of measuring color absorption or photonic crystal transmission efficiency, we're measuring the fluorescence spectrum of whatever enters this optical fiber. Okay, so this just shows our calibration. Again, so just to prove to you, here's like the spectrum from a broadband light source taken with the camera. Here's a green laser. There's a red laser going into the spectrometer. And then the type of experiment we performed here is something called a fluorescence resonant energy transfer. Maybe I'll use this next one to describe it. So the assay that we're seeking to perform is detection of a specific DNA sequence, actually a specific RNA sequence that's specific to a strain of bacteria that's resistant to antibiotics. And so people in hospitals are very interested in detecting presence of antibiotic-resistant bacteria because it can't be treated and is very dangerous to patients with compromised immune systems. And so being able to find and identify the presence and location of antibiotic-resistant bacteria is a kind of thing that would be of a lot of interest for a mobile test. So how would you do that in a rapid and sensitive way? Technology not developed by us, but published by others, is something called a molecular beacon. It's really ingenious. What they've done is to take a nucleic acid molecule that's specifically designed to have like two legs where the two sides of the DNA are held together in a double helix. But there's a loop where a nucleic acid pieces are like a single-strand DNA, not bound to anything else. So we have a specific DNA sequence that can be displayed in this loop, and then the two legs are held together. What they do is to put a fluorophore on one foot, I guess that would be like the left foot, and then put a quencher to that fluorophore on the right foot. So as long as these two things are together, this fluorophore can't give off any light. However, if you have a DNA sequence or an RNA sequence that's a complementary match to this sequence inside the loop, it will bind there, and the stress of adding that piece of DNA will cause this molecule to open up. The left and right foot will be separated from each other, so the quencher gets removed from the fluorophore, and now we'll have brightness. And so basically, you have this floating around in liquid. If the specific DNA sequence is present, the liquid will go from dark to bright when illuminated with a laser where it can give off fluorescence. So just to give you an idea how dramatic actually this kind of reaction is, here's our molecular beacon sequence. It recognizes a complementary sequence. This is a reaction that takes place in liquid. It's called a homogeneous assay. If we illuminate a sample with just a molecular beacon, here you can see the green laser showing up a little bit in our spectrum. Here, if we have 1 micromolar of the DNA, so that's a lot of the DNA actually, we see the fluorescent spectrum being given off, and there's still our green laser. So this is what we're essentially looking for. So how well do we do with this test? So we did some tests with a perfectly matched DNA sequence, and we did some other tests where we took only one base and changed it to see how selective it was. Here just shows the matched DNA sequence measured with the phone. So here we're measuring the fluorescence output. So this is not absorption spectrum anymore. This is like actual, like positive value means more light. For high concentration of the specific RNA sequence, lower concentration, and really lower concentrations. If we zoom in on the really low concentrations, we can see like a little bit of airspace, between 0 concentration and 1 picomolar. I think that's the blue one here. And so if we measure these same exact liquids, actually take the same liquids and put them into the Agilent Fluorimeter, this is what we measure. So we see the spectrum, but if you go down to the low concentrations, you can't even tell the difference between the several lowest concentrations. So in fact, with our smartphone, we're outperforming the laboratory instrument. And so if we take the area under that fluorescence spectrum, integrate it, and make that into a fluorescence emission number, we can measure the fluorescence intensity as a function of the RNA concentration. And so this is just our dose response curve. The 4 lowest concentrations are here, blown up, so you can see we can see the difference between 15 picomolar sample and 0. So our limit of detection around 15 picomolar, which is pretty respectable for a smartphone. This isn't going to detect like a single strand or anything, but 15 picomolars is fairly low. And in fact, with the fluorimeter, the 4 lowest concentrations can't even be distinguished from each other. So in fact, we're doing a little bit better. Let me ask if anyone can guess why our smartphone worked better. If anyone has used a fluorimeter before. Any wild guess? Yeah, so the reason is actually the light source. We cheated a little bit, and I didn't tell you how we cheated. Is that with our phone, we're using a green laser pointer. So green laser is pretty intense, and we focus that light down right into the sample and using a fluorophore that absorbs green light very efficiently. The fluorimeter uses a xenon lamp. It's more of a general purpose laboratory instrument that's meant for exciting many different fluorophores. And so the xenon lamp has specific wavelength bands, many of which don't excite the Psi3 fluorophore at all. And so really its excitation at the absorption wavelength of the fluorophore wasn't as efficient. But who cares? We still did better than the fluorescence instrument, and actually that was our goal anyways, is to show that the phone could do pretty well. So this just shows the mismatched sequence, just to let you know that our test is very selective. If we just change one base pair, or one base out of that strand of detected RNA, we get like no sensing at all until you really overwhelm it with a lot of stuff, and you start to get some binding. So the interaction is actually very specific. That has more to do with the molecule than it does to do with us. So going forward, what we're doing with this, we did a lot of work with this iPhone 4 and this aluminum device just to show what it could do. But then it wasn't long before the iPhone 4 was old news, and there are lots of other great phones coming out. We found actually that the iPhone platform is kind of limiting in terms of what you can do with it, with controlling the phone itself. It doesn't let you get into the settings to control things like exposure time even. So we've made transition to Samsung phones, even though I still stick with Apple phones in my personal life. But what we've been doing is something a little different that allows us to interface more easily with different phones, because every phone's got a different camera, different LED light source. So in fact, what we're using now is the internal flash LED as the light source. We take an optical fiber, just put it right up against the LED. So the light from the fiber is what comes out and illuminates our sample. If we're doing an ELISA measurement, we shine light through the sample. It gets bounced off a mirror and comes back into a second fiber. Then now the second fiber acts kind of like the pinhole I told you about before. And then that light goes through the same collimating lens, same diffraction grating that I told you about before. But basically, with these two optical fibers, we can interface with any phone pretty easily. And then for doing fluorescence measurements, we've built the laser pointer into the detection system. I show it as a big laser pointer, but it's actually just a small diode. Yeah, and this is what it looks like. So now we're making these out of 3D printed plastic. So you see actually most of it is airspace where it has the optical fibers going in. The phone just slides right into it, puts the back-facing camera in contact with the diffraction grating. There's a battery for operating the green laser, but that's our system now. The cartridges that we measure slide into the back. There's a slot in the back that we put the device in for doing measurements. Let me skip through this. I wanted to show you one more thing, just because I think it's another interesting way that you can use a phone. Again, it's pretty simple. There's not like super high like science in this, but it's for doing something called thin layer chromatography. And so, in this case, we're considering applications in detecting and screening inauthentic pharmaceutical products. And so there are, I'm sad to say, unscrupulous people in the world. And so especially in developing countries, there are people who will make and sell drugs that are packaged and formatted to look like the real thing, but actually are not. So a person may think they're getting a anti-malarial drug or an anti-TB drug or an anti-cancer drug, but their pill has been diluted by 50% or substituted with inactive chemical compounds. And so we have a company that's funding us who manages the supply chain coming from the United States and Europe to drug distribution in Africa. And so they're interested in having a mobile testing capability where they can test drug authenticity at the places where the drugs are being given to the patient, like at the clinic. And so if you kind of like look at this, for example, like one of these is fake, one of these is real. I mean, they look the same. They're the size and color, that the printing on them is almost the same. And this is a serious problem. I've been told that hundreds of thousands of people killed every year because their treatment didn't work as it was supposed to. And so this is really a serious problem. Oops. Yeah, and so this is like a place where we take our like Walgreens or CVS pharmacy for granted, but if you're buying drugs, in some places it looks like a flea market where there's just things on tables, people picking through them. Who knows if any of it is the real thing. So the approach that we're seeking to use with a smartphone is something called thin layer chromatography. The reason that we're using it is because it's really simple, very inexpensive and simple procedure to perform. What you do is there's a aluminum card. It's about the size of a microscope slide, maybe 1 inch by 3 inches. These are plates made out of aluminum that are covered with a nitrocellulose paper coating. What you do is to place a droplet of the sample you want to test on a horizontal starting line. Then you take this card, put it into a plastic container, close the lid. The plastic container has a solvent in it that gets drawn up the card by capillary action and drags the drugs, whatever's in these spots, along with it. And so after the solvent front reaches a certain point, you just open the lid, you take the card out of it, and so now your drugs are somewhere between here and the end. The nitrocellulose material is infused with a phosphor that glows green if you illuminate it with ultraviolet light. And so the presence of a drug or some material inside that phosphorescent material will quench the phosphor. So instead of it being green, you'll see a dark spot on a green background. So that's what we're looking for, actually. We're trying to measure something that's green that has dark spots on it. And what we want to do is to measure the vertical position of the spots to tell us like what the drug is. And we also want to measure the darkness of these spots that helps make an estimate of the concentration. Okay, so the way that we do our testing is we have always 3 spots, only 3. Maybe someday we'll expand to 4, but right now it's only 3. We would have the left spot and the right spot would be known concentrations of the drug. So one spot, I think we usually use the left. Here I said there's 4 spots. There's really only 3. So one of the spots would be 100% concentration. Another spot on the right would be a lower concentration, say like 80%. And then our drug under test would be the center one. So we have like a built-in calibration standard in this test. And so if you were to see, so if it's a single component drug, you should see 1 spot. If it was a 2-component drug, you would see 2 spots going up and down. If it was supposed to be a single component drug, but someone added something to it that was not the drug, then you'd see 2 spots instead of 1. That's the basic idea. And so what we did was actually pretty simple, is that we made by 3D printing a plastic housing where we could take a phone, pop it on top so that the phone's camera looks down and is focused on a TLC plate that gets slid into a slot on the front. This cradle holds a UV illuminator, just a battery-powered UV lamp that goes in and shines light at the sample, so making it glow green. While this takes a picture of the green light, and then we have some software, an app that we wrote that analyzes the position of the spots and estimates the darkness of the spots. And so our first, so this whole thing actually costs like less than $100. We're the most, actually not counting the phone. But since this is for in Africa, actually, they instructed us to use like an old, inexpensive, crummy phone. So this is like a 1 megapixel camera phone that you can get on eBay for $100. So we didn't want to use like fancy phones in Africa. So this just shows like one of the actual images, full strength, 90% strength, and 80% strength of one drug. This is a malaria drug, where the software determines the position of the horizontal line, and all it does is measure the vertical travel distance and the spot depth or darkness of the spot. So I just wanted to share some of our first results on this with you, just showing you there, if I can get this. This isn't the best. I've been happier with my other laser pointer. Uh-oh. Oh, my technology is backfiring. Oh, I have a message. Ha! I have an appointment in 15 minutes, so apparently, okay. Ha! Okay, I have to go back now. I'm near the end. Okay, so this just shows some of the results, measuring the spot density. All right, so we took our phone, took these measurements. There's a lab instrument called a densitometer. It's a $50,000 instrument that does kind of the same thing. It illuminates the slides with UV light, takes a picture and analyzes the image. Maybe we'll put those guys out of business. I don't know, but we use a densitometer to measure the same plates, to measure the concentration and the travel distance for spots in a TLC plate. And so the red is a densitometer, the black is our smartphone-based measurement, and it basically measured the same thing. And we could easily tell the difference between 100% concentration, 90% concentration, and 80% concentration. And we've done this for several drugs. Let's just show you a few examples here. So nevirapine is a different drug. Paracetamol, yet another one. So believe me, we've done this for lots of drugs now. So just to conclude, I think that we were showing that the internal sensing capabilities of phones is sufficient, that they can do some of the same work, deliver the same detection sensitivity, the same resolution for doing many types of biological assays. Any assay that generates fluorescence, any assay that generates a color change of a liquid, any assay that has a biosensor with some kind of resonance in it, we've shown that we can do this way. And so we're kind of working on this capability now for all kinds of assays. So we're working now on an approach for doing infectious disease diagnostics for horses and other animals to identify respiratory diseases that are easily communicated among like the herd. There's a lot of applications in people as well, but the animal ones are easier to do first because we don't need to have so many approvals and stuff like that in order to test them. So hopefully you'll see more and more happening in this area. A lot of people are catching on to the fact that smartphones can do a lot. So finally, I just wanted to acknowledge my students who did all the hard work that I showed you here today. So they're in majors, everything from bioengineering, electrical and computer engineering, material science, and even the physics majors are in my group. We've been fortunate to have some great collaborations that help us on the software development. Professor Lumeta's group and Ukan Demirci's group at Stanford was the group we were collaborating with for the HIV detection. So thanks so much for your attention. I'd be happy to answer questions.
So quick questions? So for your thin layer chromatography, is it practical is it planned to screen the drugs, each drug that each person is taking each time? Or is that like not really practical? How practical is that?
So what they're seeking to do is to have this be a screen that's performed at the clinic where the drugs go from like the doctor to the patient, like the last link in the chain. And so what they're seeking to do is to have some quality control in the clinic. We don't expect like the patients themselves to be testing like each of their drugs, but this would be making sure that nothing happened along the way, like through the supply chain on the way to the clinic. So like strange things happen. I've understood. Is not like artificial drug distributor will show up and here's your paracetamol shipment for this week, and so they want to verify like in the last step at the clinic. And then what we're doing actually with our app is taking photo of the pill, photo of the package, serial number of the package, and all that information is going back to a database where people will try to monitor for like the presence of inauthentic drugs and to match them up where they occur in other places, and as a way of maybe even doing some backtracking detective work to find where the bad guys are. So part of it is the measurement, part of it is just like the information system to help track where where the inauthentic drugs are being distributed.
But how about the picture of flea market?
Yeah, they made.
How is that like? People are still buying from there, right? So it's like.
Yeah. So I think it would be dangerous to buy your drugs at the flea market, and if you did, maybe the flea market guy should get one of these, and if they want to, convince their patients that, or their customers that things are real, that they've tested them. So.
Just one quick.
Yeah. So great, so quick question is, do you think this is a technology that will advance as phones advance as well? Or I know for the case with the densitometer, that wasn't the case because you guys were limited on the type of phone to use. But with all the other examples that you gave, do you think in the future you'll develop new software, new program for the new phone that's coming out, new software that's integrated in the phone?
Yes, yeah. So I think the same way that anyone who makes accessories that go with phones have to stay updated, and so every phone model is going to have the camera in a slightly different location. The flash LED will be in a slightly different location. Our approach is flexible, that you could make a cradle that puts the optical fiber in the right place for a particular model phone. But like you say, it's like an arms race. They're always going to be evolving. But I think actually the evolution of phones is they always keep getting better. And so I think that's actually fortunate for us from the standpoint that the better the cameras get, the more processing power they have, the better our image processing and spectral resolution and all those things will get. And it's kind of like a flea on the back of a giant elephant. We're just kind of going along for the ride and taking advantage of all these technical developments that happen with mobile devices.
So Brian, have you worked with your favorite company on this?
My favorite company?
Yeah.
Apple?
Yeah.
Yeah. So Apple is they're like a tough nut to crack. And so they hired one of my PhD students who now works there, but she won't tell me anything about what they do. So they're interested in mobile health, but I think they're interested in like what they can put on the Apple Watch. But we do have interactions with a collaboration with Motorola and with Foxconn on these things. They're interested to see where it'll go.
Would iPad be a potential usage?
I think the same thing with the iPad or tablets as well. Yeah, you just have to have a different thing that just interfaces with the camera and the flash on those devices as well.
We 1 minute, because you guys have an appointment.
Oh, I'm fine. I got 5 minutes to get upstairs. So.
Yeah, your smartphone can distinguish or detect how pure it is. A seed plot, for example, different seed like weeds inside the soybean seed lots. It can be managed to do variability or to do distinguish between different seeds.
And so the question is whether we can distinguish between different types of seeds. I don't know. We haven't considered that before. And so I guess my answer would be if there's an assay you can perform in a lab that generates fluorescence output or a color change in a liquid, and those are the kind of things that we can do. So people have asked me that actually, is like whether we could detect the genetically modified crops, or if their food had a genetically modified crop in it. And I don't know the answer to that. I mean, so it's a specific DNA sequence. So I suppose if you had like a PCR test that generated fluorescence output if a specific gene was present, like the anti-Roundup gene or something, that you could imagine doing like a mobile test like that. But it's like the tester and the assay both have to go together. So it could be very similar to the way we're detecting pathogens with like amplifying output of a specific DNA sequence that has a fluorescent output. But we haven't really considered like those kind of applications yet.
Okay, so we thank now Dr. Cunningham for his
Thank you.
Thank you for keeping your questions short.
Thank you very much.
Hi.
So like for the peanut allergy.